Good afternoon. I'm delighted to be joining you in this global virtual symposium on AD genetics. And I thank the Alzheimer's Association, the NIA, and the ADSP for this chance to speak briefly on exciting genetic research emerging from Asia and Australia. Asia is Earth's largest continent and has 60% of all humans. So obviously there's a lot of genetic diversity, exacerbated by the fact that there are these features like the Himalayas, resulting in different regions in Asia. Some of this genetic diversity has been explored through a project called Genome Asia that's ongoing. And pilot data from the first 1,200 were published in Nature last year, studying ethno-linguistic and caste groups, about 219 population groups, and showing that there are over 10 ancestral populations underlying the people in Asia versus about 1 for people of European ancestry. What does that mean? That of course means that there are founder effects. And so a larger number of loss of function mutations that are novel are likely to be found by studying this population. Today, we'll explore what's happening with AD and related disorders genetic research in just 2 areas, South Asia and East Asia, with some Australian efforts. So, in South Asia, a large region, you find that there are many groups with founder effects. All of these groups have higher than in the Finns, who have the highest IBD in Europe. And this is not just isolated tribes. In the city of Chennai, that's as large as New York, IBD is 40% higher than in the Finns. So it's not surprising that among the over 150,000 loss of function variants that Genome Asia found in its pilot, one of them was in the gene ABCA7, where we know that loss of function variants can increase risk of Alzheimer's. So this kind of human knockouts are a great way to understand the phenotype of the gene. So studying Asia, India, are several genetic efforts. India is a country with over 22 languages and many dialects. It's a very young country with a short lifespan, although the lifespan is rapidly increasing. And because of the size of the population, there are a large number of older adults. I'll just touch upon 2 of these studies. One out of Bangalore, the Silicon Valley of India, has both an urban component, somewhat like the BLSA, a very highly educated group of people who have a high risk of hypertension, undergo very detailed testing, including MR, and a rural sample in the county or taluka of Srinivasapura near some old gold mines. These people are on an average 9 years younger, have only a middle school education and less hypertension. Despite that, when you look at the MRI from these 2 groups of people, the rural people with lower hypertension actually have more white matter hyperintensity done on the same MR machine in the same way than the urban ones. What could be the reason? Part of it could be genetics. So Genome India is an effort where this Bangalore site, the Center for Brain Research, is one of the sites. There was a talk by the head of CBR, Dr. Vijayalakshmi Ravindranath, on the last day of the AAIC as a plenary talk. And those of you who are interested can learn more there. But this is a plan to sequence about 10,000 people across 4 sequencing centers, 22 institutions, to study the over 4,000 ethnic groups in India because of things like waves of invaders as well as endogamous marriage practices. All this and other genetic efforts in India are going to help us better understand the genetics of this subcontinent, just like Genome Netherlands and Iceland help us design better chips. Some of the strengths that India possesses are highly trained neurologists and neuropathologists, but some of the challenges are the many languages being spoken and the low education. Another effort, modeled more after the health and retirement study, led by Dr. A.B. Day at the All India Institute of Medical Sciences, Dr. Jin Cookley in the University of Southern California, and colleagues at University of Michigan, studies people from multiple universities across this diverse subcontinent. Over 72,000 adults are studied using economic background. And then among the older adults who are oversampled, a subsample undergoes detailed assessment for dementia. This subsample is enriched for those most likely to have cognitive impairment. And then they undergo a more detailed cognitive assessment as well as a consensus dementia review by experts. From among these 4,000 people, about 1,000 have had GWAS, and about 3,000 will go on to have whole genome sequencing. Results of this GWAS show that there is a large proportion of ancestral South Indian group that's not related to any other known genetic group. And that the known AD polygenic risk score explains very little of the variance in cognition. However, there are variants that are going to be found as seen in this QQ plot. And so MedGenome will do whole genome sequencing. It will be QC'd at UPenn and shared through NIAGADS. What are some preliminary results? When one looked for associations in the known AD genes, obviously 1,000 people is a small sample for genome-wide discovery. But in these 43 genes, after correction for multiple testing, you find some of them are indeed associated with the risk of worse memory performance. One of them is ApoE, but it's not the traditional E4 SNP, it's another SNP, which also changes the protein structure of ApoE. And then there are other genes that where the mineral allele frequency is quite different in Indians as compared to persons of European ancestry. Looking at a more complex general cognitive factor derived by Alden Gross and colleagues at Johns Hopkins, you found some novel genes, including DDAH1 that's involved in synthesis of nitrous oxide, that's a vasodilator, and STAP1 that's involved in cholesterol metabolism. Moving now from South Asia to East Asia, in Japan, one of the landmark studies is the Hisayama study, ongoing since 1961, with exceptional follow-up and over 75% autopsy. And it's found that there is, contrary to North America, an increasing prevalence of total dementia, particularly of Alzheimer's, although the prevalence of vascular has not changed much. And looking at this population, some papers have shown that, for instance, in Japanese, there's an interaction between E4 and PICAM gene. In E4 negative persons, PICAM does not increase the risk of AD, but when present together, there is a 600% increase in risk. Similarly, though, when one looks at risk determinants among ApoE negative people, you find that this gene CNTNAP2 emerges, and this has a protein product, Caspr2, that's expressed on the synapse and associated with risk of a range of neuropsychiatric illnesses. When one looks at Korea, there is a large cohort at Gwangju where in collaboration again with Lindsay Farrer and others at Boston University, there have been genetic publications. Dr. Lee and colleagues see an amazing 12,000 people each year, about half of who undergo the full assessment, including CSF and PET, and a sixth of whom are on follow-up. And they found, for instance, that there are variants in ApoE that are more common in East Asians, and they're in the promoter region, alter the amount of ApoE protein synthesized in the brain, and modify the effect of E4. Thus, if you have a variant in that gene and you are an E4 carrier, then having a TT variant lowers levels of ApoE and increases risk, also decreases age at onset, results in smaller cortical thickness. But with the E33 or 34 heterozygous, this variant doesn't have much effect. Coming to Australia, there are a number of efforts. In Perth, Nicola Armstrong and colleagues worked with the CHARGE consortium to show that periventricular and deep white matter have different genetic bases. This is in Queensland. Sarah Medland and colleagues worked with Enigma Consortium, UK Biobank, and CHARGE to explore the genetic basis of 34 regions of interest in over 50,000 people, and found that genes that determine prenatal cortical development are important, that surface area genetically correlates with general cognitive function. In Sydney, Henry Brodarty, Parminder Sachdev, and others have put together an Asian cohort of studies of memory, and working with a European JPND Bridget consortium have looked at genetics and epigenetics, including whole genome sequencing. In Melbourne, my colleagues Matthew Passe and others, Rachel Buckley and others are collecting salivary DNA on over 10,000 people across Australia through the Healthy Brain Project that assesses cognition through computerized testing. And also in Melbourne, Paul Lacaze and others are obtaining genetic data on the large US-Australian ASPRI study of aspirin for prevention of dementia. And over 3,000 people from this ASPRI group will have whole genome sequencing. About 13,000 have had GWAS, and some preliminary papers on polygenic risk scores have been submitted. For a flavor of other efforts across Asia, there are biobank type studies like the China Kadoorie study in conjunction with Oxford and modeled after the UK Biobank, sampling people across 10 regions in China. The Japan Biobank, which is more like the BioView or BioMe studies, starting with clinical data and then obtaining genetic data on over 200,000 people. There are other cohorts that might be for a specific reason, like the Japan ADNI study or the worldwide finger study on which DNA is being collected, as well as a number of studies that have, say, an endophenotype or imaging focus, like the Indo-Dutch study out of Delhi and the Rotterdam study. So I'd like to end by thanking all of you, and I welcome questions.